The overall goal of this Ex Ovo banding procedure is to create a reproducible, non-lethal alteration in cardiac hemodynamics to study the resulting genetic consequences.
This method can help answer key questions in the cardiovascular development field, such as the development of cardiac valves and associated disease states. The power of this system is that it can be used to study genetic pathways that respond to changing hemodynamics in the developing heart and valves, and also in help finding a tipping point at which rescuing the heart does not have any long-term deleterious effects. So demonstrating the procedure will be Lorain, Junor, a technician from my laboratory, and Mr. Vinal Menon, a PhD student in the laboratory, who will be demonstrating the handling of the eggs and the use of the ultrasound machine.
Incubate 80 to 90 fertilized bovine chicken eggs with their blunt ends up on plastic egg trays in a humidified rocker incubator set to 40 degrees Celsius. Let them develop for approximately 72 hours to HH17. Once at stage 17, pour about 20 milliliters of warm Tyrode's buffer, supplemented with sodium bicarbonate, into a 100 millimeter by 26 millimeter dish. Next, remove an egg from the incubator and sterilize it with 70% ethanol. Then, gently crack the shell using a scalpel handle and carefully release its contents.